At 42.7, adaptations for gas exchange include pigments that bind and transport gases. So now we're shifting gears into what is directly responsible for transporting the oxygen and carbon dioxide to the various places within the body. And again, we're really thinking about the role of metabolism here. So metabolic demands require that blood be able to move large amounts of oxygen and carbon dioxide. Here we'll walk through the process of circulation and its relationship with gas exchange. So starting on the right-hand side, we are taking in air. We're able to do that due to changes within the thoracic cavity that allow for air to head down their partial pressure gradient. Now that air will make its way into the alveolar space, and then the oxygen will go down its partial pressure gradient, so it will diffuse from the alveolar space to the alveolar capillaries, which have a very close spatial relationship. So at this point, we now have our vessels that are going to have increased concentrations of oxygen, and those pulmonary veins are going to make their way back to the heart. And we're on the left-hand side of the heart, so we have our left atrium. Our blood's going to drop down to the left ventricle and then eventually be pumped out through the aorta. And then that blood that's oxygen rich will head out to the rest of the body, and that is systemic circulation. At systemic capillaries, we will have an exchange take place because at the body tissues, these will be sites where work has been done and ATP has been generated, metabolic waste has been made, and now we're saying we need more of that final electron acceptor. So we're going to take on some oxygen here that's coming from these oxygen-rich vessels and drop off the CO2 that was produced through carrying out our everyday metabolic actions. And now we have oxygen poor blood that will make its way through the body, ultimately heading towards pulmonary arteries, and those pulmonary arteries are going to head towards the alveolar capillaries. And at this point, we're oxygen depleted, but we're carbon dioxide rich due to the metabolic activities taking place of the body tissues, and we want to get rid of that. We don't want carbon dioxide to build up within the body. So that carbon dioxide will diffuse down its pressure gradient, heading into the alveolar space, and then from there it can be exhaled. Now, within this process, there are technically two sites of respiration. There's external respiration, which is the pulmonary gas exchange that takes place. And then there is internal respiration, which is the gas exchange that's occurring here at these body tissues. So this is just connecting the earlier content of blood flow without as much detail as what was discussed at that point, but then connecting it with the components that are associated with respiration as well. This image is similar to the one that preceded it. The only thing is that it adds on the actual values of pressure seen for our gases of oxygen and carbon dioxide. Now, the actual numbers are not what is essential for us. What I want there to be focused on is the fact that the partial pressure gradients exist, and that is what drives diffusion from one location to the other across the respiratory membrane. So we're just looking for confirmation that the difference is what is driving movement from one site to the next. Now, when it comes to the oxygen gradient, just one thing to notice is that that's steeper than the carbon dioxide gradient, but equal amounts of the gases are exchanged. And that's just due to the difference in solubility of carbon dioxide. That's 20 times more soluble in plasma and alveolar fluid than oxygen is. So we do see a difference between oxygen and carbon dioxide with respect to the gradient, but that's just due to a difference in solubility. So your take home is that these values and the differences that we see as we migrate from one space to the next will let us know the direction that our gases will flow. And the fact that we see a stark contrast between the gradient for oxygen versus carbon dioxide has to do strictly with the level of solubility of the carbon dioxide gas compared to oxygen. So we'll just focus on hemoglobin as a respiratory pigment. And hemoglobin is a polypeptide that has reached its quaternary level of structure because there is 2 or more polypeptides involved in it. It consists of 2 alpha chains and 2 beta chains. And each chain is conjugated with a non-protein heme group. And the heme group is what binds oxygen to an iron atom at its center. So, with that said, if each heme is carrying 1 molecule of oxygen, a hemoglobin molecule can carry up to 4 oxygen. So, the maximum amount that a single hemoglobin molecule can carry is 4 oxygen at a time. So, here we have hemoglobin dissociation curves, which is a way to think about the relationship between hemoglobin and oxygen. Now, hemoglobin has those 4 polypeptide chains. They have their iron-containing heme group, and they can interact with up to 4 molecules of oxygen. When it comes to oxygen loading, it's a rapid process and it's a reversible process. When you have hemoglobin and oxygen combined, that's referred to as oxyhemoglobin. And when hemoglobin releases oxygen, it's referred to as deoxyhemoglobin. Now, after the first oxygen molecule binds to iron, the hemoglobin molecule is going to change shape. And as a result, it more readily takes up additional oxygen molecules. And that helps to enhance the saturation state. And this is comparable to a concept that's discussed in Bio 100, which is cooperativity, in which the binding event of a substrate is going to enhance the other subunits and their ability to interact with substrates at allosteric sites. So this is an opportunity to connect that older content with our current content. So here we have hemoglobin, essentially oxygen hemoglobin dissociation curves, which helps us to understand the relationship between hemoglobin and the partial pressure of oxygen in varying locations. So if we look at the axes, we have one axis that tells us the partial pressure of oxygen dissolved in the fluid that is proximal to the hemoglobin, surrounding the hemoglobin. And on the y-axis, we have oxygen saturation. So if we're at 100% oxygen saturation of hemoglobin, that would mean there are 4 bound oxygen molecules. So let's take a look at the different locations in which we have our hemoglobin. So if we are at the lungs here, then what we are saying there is that the partial pressure of oxygen is high and hemoglobin is pretty much fully saturated. And that's just based on where it hits kind of the graph here. So not quite at 100, maybe 99% saturated at this point. Now, if more oxygen is present, more oxygen is bound. Now, when it comes to the process of the strength of that binding, the idea of cooperativity works in both directions. So just as readily as it will pick up additional oxygen molecules in oxygen-rich locations, as it starts to drop off oxygen, there will be a really swift change in saturation. So that's why there's a more sigmoid curve that's seen within these dissociation curves, which is the type of trend that we also see for enzymes that display cooperativity. Now, if we are in tissues of other organs where the partial pressure of oxygen is lower, then hemoglobin becomes less saturated. And if we are at locations like tissues during exercise, we expect the least amount of saturation at this point because those are areas that really depend on oxygen in order to continue going through those events. And they're also depending on the ability to release their carbon dioxide too. So here we have different examples, so lungs and tissues at rest, tissues during exercise, and then we're thinking about how the saturation of hemoglobin shifts in these different locations. And the binding strength or the way in which those shifts occur is going to be influenced by the cooperative nature of hemoglobin-oxygen interaction. On the right-hand side, we have a dissociation curve that thinks about the relationship of pH of the blood with the amount of oxygen that will be found bound to the hemoglobin protein. And as a reminder, under standard physiological conditions, pH should be about 7.4. And here we have an example where our pH goes down to 7.2. And what we see here is that our level of saturation at the same amount, with the same amount of kind of partial pressure, but varying pH conditions, we don't have the same exact amount of oxygen bound. So we are going to see a little bit less oxygen that's bound when the pH is lower than when we are at normal pH conditions. And I'm going to go over several factors that influence oxygen unloading, but this would be an example of the Bohr effect. So when we have decreased pH, that causes the dissociation curve to shift to the right, which is the solid line, and that response is the Bohr effect or the Bohr curve. So, as was evident in the dissociation curve on the left of the previous slide, hemoglobin doesn't unload the same amount of oxygen to all tissues. And so we saw the difference for tissues at rest versus tissues during exercise. And so there are several factors that influence the rate at which the dissociation event's going to occur. And that should make sense even without utilizing a dissociation curve. So at the top hand side of this, we have normal conditions, so normal body temperature, proton concentration, or partial pressure of carbon dioxide. And, honestly, as far as the illustration goes, it's only as good as kind of overlaying a difference, which you can see in this bottom portion. Now, let's go through the different factors. So one is temperature. So temperature influences unloading because what we find is that active tissues are warmer than less active ones. Thinking about metabolic processes, there is a release of energy that cannot be used, and it is released in the form of heat. And so we find that active tissues are warmer than less active ones, and that results in extraction of more oxygen from blood passing through them. So increased temperature increases the amount of oxygen unloading at that site. When it comes to pH, we can kind of consolidate panels B and C because when it comes to active tissues, they generate extra carbon dioxide, and extra carbon dioxide raises the concentration of protons when we think about how carbon dioxide interacts with water, and that ultimately lowers the pH. So if carbon dioxide in water produces carbonic acid will dissociate into bicarbonate and protons. And this event weakens interactions between oxygen and hemoglobin, and it is more pronounced in systemic capillaries than in the lungs. And this is the Bohr effect that was mentioned in the previous slide. Now, all of these factors are going to cause more oxygen unloading. And these factors are going to influence the three-dimensional structure of the hemoglobin protein. And as they do this, they're going to lower the affinity or attraction that hemoglobin has for oxygen. So if we look at the graph on the top versus the graph on the bottom, we see that these events will cause the dissociation curve to shift to the right, just like the earlier slide. So these factors are usually highest within the systemic capillaries, and that is or should make sense because these are locations in which the goal is to release the oxygen molecules. So collectively, these events lead to the Bohr effect that helps to enhance oxygen unloading by weakening the desire for oxygen to stay associated with the hemoglobin protein. So let's begin with the movement of carbon dioxide within systemic circulation. So we'll start at the mitochondria. This is the site of aerobic respiration. And aerobic respiration is going to be the breakdown of organic fuels through the utilization of oxygen. And this drives the production of energy in the form of ATP. But in addition to creating ATP, it's also going to generate byproducts, byproduct being carbon dioxide. So there's a buildup of carbon dioxide within tissue cells. This carbon dioxide will diffuse down its partial pressure gradient out of the tissue cells and into the systemic capillary, where it can be carried within the bloodstream. And it can do this in a few different ways. It can be dissolved in the plasma. It can be chemically bound to hemoglobin. And when it comes to this binding event, the carbon dioxide does the binding at the amino acid location of the hemoglobin rather than the heme. So carbon dioxide binds to the globin, oxygen binds to the heme. So there's no competition with respect to transport between these two gases. And the third most popular or kind of most readily utilized would be as bicarbonate ions in the plasma. But we need to be able to convert carbon dioxide into those bicarbonate ions first. So this image is showing us the process of carbon dioxide conversion. So the majority of carbon dioxide is going to be hydrated. This is going to lead to the formation of carbonic acid, and carbonic acid is unstable. And so it's going to dissociate into bicarbonate and into these protons that are here. Now, this event is being shown to you, and by this event, I mean the production of bicarbonate ions within the erythrocytes, but it can also take place within the plasma. However, the erythrocytes contain the enzyme carbonic anhydrase, and that helps to catalyze this reaction more readily than what would occur within the plasma. The protons that are released from this process of dissociating carbonic acid are going to bind to hemoglobin, and this contributes to the Bohr effect, which enhances oxygen's release. So this means that the protons released during dissociation are not significantly contributing to the pH of blood because we're tying it up in this binding event that is occurring right here. Now, once you have bicarbonate, it needs to head into the plasma. So it needs to go from erythrocytes out into the plasma where I have drawn it. And this movement is going to be coordinated between bicarbonate movement out of the erythrocyte and chloride ion movement into the erythrocytes, and that creates a balance of ion transport. This is referred to as a chloride shift, and it occurs through a protein, so this little circle here, that is found on the erythrocytes, and it allows for antiport facilitated diffusion. So antiport, just because the movement of these ions is in opposite directions, there is no utilization of energy here. So this is not going to be active transport, but rather diffusion that's facilitated through the utilization of a protein. So we have antiport facilitated diffusion as the bicarbonate and chloride ions are transported in opposite directions. So now let's look at carbon dioxide transport within pulmonary circulation. So for pulmonary circulation, notice the change in direction that's seen through this protein that's here. So now we have bicarbonate coming in and our chloride ions leaving, because now we need to load that bicarbonate back into the erythrocytes so that we can convert it back into the form that it will take as it exits out of the bloodstream and into the alveolus, which will be in the form of carbon dioxide. So once we're in the erythrocytes, we have bicarbonate, which combines with protons, and that will form carbonic acid. And we're utilizing carbonic anhydrase again, which will serve to split carbonic acid into water and carbon dioxide. So we have our carbon dioxide release. So this reaction frees up carbon dioxide, which can then diffuse down its gradient into the alveoli. So again, along its partial pressure gradient. Now, the amount of carbon dioxide transported in blood is affected to the degree of oxygen that is found there. So, as a reminder, this is not due to competitive binding because oxygen and carbon dioxide do not bind hemoglobin in the same locations. This phenomenon, however, is referred to as the Haldane effect, and it reflects the greater ability for a hemoglobin that contains less oxygen bound to it to form carbaminohemoglobin. So as carbon dioxide comes in from the systemic bloodstream, its binding causes more oxygen to dissociate. And that's for reasons we discussed in factors that influence oxygen unloading. This dissociation allows more carbon dioxide to combine with hemoglobin. One more thing to notice is that when it comes to this conversion back to the form of carbon dioxide, is that we're utilizing the same enzyme here. So this enzyme can be utilized for a reversible reaction. So we'll close out with thinking about respiratory adaptations that are seen within diving mammals. And this is really just to introduce the idea of myoglobin. And diving mammals, as well as us, have the ability to store oxygen within myoglobin proteins. And myoglobin proteins are essentially a type of oxygen binding molecule found in muscle cells. It has a single polypeptide chain with an iron atom that can bind to an oxygen molecule. But the difference between myoglobin and hemoglobin lies in its affinity for the oxygen molecule. Myoglobin has a higher affinity for oxygen, which means that when oxygen levels fall in muscle cells, myoglobin is going to hold on to that oxygen until hemoglobin supplies have been exhausted. And so a benefit for deep sea diving mammals is that they can stay underwater for longer periods of time because of the high levels of oxygen that's stored in the myoglobin within their muscles. And there are also other mechanisms that allow for mammals to conserve oxygen, which can be through modifying buoyancy, rerouting blood to areas that need it, or deriving ATP in muscles from fermentation once oxygen is depleted. So a lot of concepts apart from the buoyancy that we've already previously discussed. So a really important take-home message is that difference in affinity between myoglobin and hemoglobin and how that would impact dissociation curves for one over the other.